Hi, uh, I'm Vivek Patni, and I'm going to discuss about the rapid phenotypic identification of the microbes. So, this rapid identification, basically helps us to reduce the time used for the identification of the bacteria. So, what is basically phenotypic identification, which basically includes structural characteristics, biochemical properties, metabolic capabilities, serology, as well as the fatty acid analysis. Microscopic morphology, which basically defines, you can say, examples such as uh, shapes of the bacteria, size, okay, cultural characteristics, what are the which type of colony does a bacteria develop, what is its the size of the colony, and what is the color of the colony, metabolic capabilities such as let's say fermentation of a particular compound, enzymes produced by it, serology, serological varieties, and fatty acid analysis. And why do we need the rapid phenotypic identification? So, reducing the time taken for the identification and antimicrobial susceptibility testing basically contributes to improving the care of the patients with the infection, especially in the context of increasing the antimicrobial resistance. The phenotypic methods offer a huge advantage over genotypic methods in that they can identify a wide range of types and detect the resistance currently expressed and resist the genetic variability in the resistance associated detection. So, to some points, why do we need the rapid phenotypic identification? Less time is required. Check on resistance of the antibiotics, reduces the human errors, increases the productivity, more number of tests in less time, and operate with fewer instruments. There are also some disadvantages also, such as the automation, basically which requires automation is basically expensive for microbiological laboratories, need pure cultures because the mixed cultures will pick up only the dominant ones, or you can say the mixed results also. Antimicrobial susceptibility sometimes may not be possible, does not help in the identification of the viruses. It identifies only those organisms which are present in the database. So there are some systems in the market such as Biolog, API system, Vitek, BD Phoenix, and Celoscope Technologies Bioscience Solutions. In this video, basically I am going to discuss about the Biolog system. So the Biolog system, Biolog's powerful carbon source utilization technology accurately identifies the environmental and pathogenic organisms by producing a characteristic pattern, or you can say metabolic fingerprint, from the discrete test reactions performed within the 96 well plate microplate. Okay, and the microbial this Biolog microbial ID system can rapidly identify over 2,900 species of aerobic and anaerobic bacteria as well as yeast and fungi. And the culture suspensions in this case are basically tested with a panel of pre-selected assays, then incubated and read and compared to extensive databases of environmental organisms, human pathogens, veterinary pathogens, as well as plant pathogens. These 96 assay reactions are coupled with a sophisticated interpretation software which delivers a high level of accuracy that is comparable to the molecular methods. 1 minute per sample setup is much simpler than the DNA sequencing, and the automated pattern matching eliminates the needs for the training and the expertise which is basically required in the case of gene sequence interpretation. So OmniLog, this is the machine which Biolog uses. The OmniLog plus ID system adds the testing capabilities for anaerobic bacteria, yeast, filamentous fungi, etc. The Biolog panel is easy to use and identifies 4 times more species than other alternate systems. The OmniLog systems basically fully automated, and the process of microbial identification by incubating and reading and interpreting the results from up to 50 Biolog microplates at a time. And this system software provides the directives to guide all the steps of the testing procedure for each microplate, and including the sample placement and removal and extended incubation whenever it is needed. Additionally, these microplates can be loaded and processed at any time where there is a need for the identification. Now this basically in these reactions basically the Biolog redox dye mixtures are used, in which tetrazolium redox dyes are basically used to colorimetrically indicate the utilization of the carbon sources or resistance to the inhibitory chemicals. Here is such an example of a redox dyes where oxidized, which is an colorless one, when it gets reduced it turns purple. So basically the Biolog redox dyes mixes are easy to use colorimetric reagents for assaying a variable cell number and measuring the ability of the cells to convert the extracellular metabolites into the mitochondrial reducing equivalents. The redox dye is basically a mix of Ma and Mb, which contains a water-soluble non-toxic tetrazolium reagents that can be used with virtually any type of animal cell line or a primary cell also. And the dye mixes exhibit greater sensitivity, higher signal over the background, and a large dynamic range of commonly used reagents such as MTT, XTT, MTS, and LMR blue. So the phenotypic arrays. The phenotypic arrays present the third major technology alongside the DNA microarrays and proteomic technologies that is needed in the genomic era of research and the drug development. Just as the DNA microarrays, the proteomic technologies have made it possible to assay the level of thousands of genes or proteins all at once. The phenotypic microarrays makes it possible to quantitatively measure the thousands of cellular phenotypes all at once. So this phenotypic microarray technology enables researchers to evaluate more than 2,900 phenotypes of microbial cell in a single experiment. Through the comprehensive and the precise quantification of the phenotypes, researchers are able to obtain an unbiased perspective of the effect on the cells of genetic differences, environmental change, and their exposure to various drugs and chemicals also. These phenotypic microarrays also helps us to correlate genotypes, which phenotypes determine cells' metabolic and chemical sensitivity properties, discover new targets for the antimicrobial compounds, and can also help us to optimize cell lines and culture conditions in a bioprocess development also, and also characterize cell phenotypes for taxonomic or epidemiological studies. So this is how the phenotypic microarray looks like. Okay, with regarding the Biolog, okay, it has 192 carbon assays, 384 nitrogen assays, 96 phosphorus and sulfate assays, nutrient simulation system, 96 osmotic assays, 96 pH assays, and around 960 chemical sensitivity assays. Now coming to the procedure for the biolog, it is very simple and has a straightforward procedure. You just need a pure culture on the agar medium and prepare an inoculum at a specified cell density and inoculate the biolog microplate with that bacterial culture. And finally, after a fixed period of time, incubate with the culture which was incubated. Now you can observe the reaction and pattern also to obtain the identification result also. And finally, with the help of the identification software known as Omnilog software, you can identify the bacteria which is present, or you can also help us to determine the chemical assays also, sensitivity assays also. So finally, the applications, phenotype microarray applications, basically with the use of microbial cells, can help us to determine gene functions, compare gene knockout mutants to wild types, compare naturally isolated strains with different genetic backgrounds also, and also help us to identify the novel antimicrobial targets by finding genes which are unique to the pathogenic microorganisms, and find phenotypes present in the pathogenic but not in the non-pathogenic strains. Also find phenotypes present in pathogenic microbes but not in a host cells. Test antimicrobial targets and drugs leads by comparative phenotyping, and determine the mechanism of action of drug leads, and also compare the phenotypic changes caused by target gene knockout versus the drug addition. Bioprocess improvement, compare and analyze the different generations of the production of the strains, QC print production strains, and can scan more than 2000 culture conditions simultaneously to optimize the growth medium for a product yield also. So basically, in the process of bioprocess improvement, it can help us to identify optimal system or optimal medium used in the bioprocess of a compound. And finally, generalized cell characterization also determine metabolic activities of any microbe, or also determine drug or chemical sensitivities. And this some of the panels can also be used for mammalian cell studies also, and can help us as responder assays also. That's all for the biolog, and thank you if you watched the video, and kindly like and share the video and subscribe to my YouTube channel. Thank you. If you have any questions, you can ask in the comment section, and also please download the Science EduTech app, and the link of the Science EduTech app is given in the description. And also we have also launched a test series for the microbiology, which helps us the students to clear various exams related to microbiology, and it contains more than 4000 questions, multiple choice questions, where they can attempt and try it out at your own pace. Also for the students who are preparing for the CSIR net, for the CSIR net life sciences, we have also option for them. We also launched a MCQ test series for them also. And to know more details about it, please download the Science EduTech app. Again, the link is given in the description. Thank you for watching the video. If you have any doubts, you can ask in the comment section. Thank you.